Great. So, welcome everyone to the Southern eDNA Society's second webinar of this year, hosted in conjunction with Women in Ocean Science. So today we have two amazing women in ocean science, Dr. Miwa Takahashi and Lara Denis-Roy. And they'll be talking about the newly launched IMOS eDNA sub-facility and also harnessing eDNA for reef monitoring, and looking at a continental scale comparison with visual surveys in Australia. Thanks for coming along. I can see, oh, hey, Ollie. So as is tradition in New Zealand, which is where I'm from, I'll start off with the karakia. This is just a, a traditional opening in New Zealand to wish us for a successful meeting. Whakataka te ho ki te uru, whakataka te ho ki te tonga, kia mā kina kina ki uta, kia mā taratara ki tai, e hi ake ana te atakura, he tio, he huka, he hohu. Tihe Māori ora. I just have a few announcements for the Southern eDNA Society before we begin. So here's a quick reminder to renew your membership if you haven't already. I believe that if you haven't, you'll be kicked off the lists or something, and you won't be getting our emails anymore. If you do have information that you want to get out there, do email Martin de Brouwer, and you can get your news through our newsletter in that way, sent out quarterly. Some other things that are going on with the society is we've got a photo contest. So if you're interested in winning a free PeerJ publication, we all know that APC charges keep increasing and increasing. So this could potentially be worth a lot of money. You need to be a member of SeDNAs, and then make sure that you share your photo to LinkedIn, Blue Sky, or Instagram, and both tag SeDNAs and use the hashtag that was sent in the newsletter. Sorry, not the webinar. And the deadline for this is the 31st of January 2026. So if you're interested getting some of your cool field and filtering and lab photos out in the world, go and post and let us know. And we'd love to see some of your really cool photos. Additionally, this is a call for the conference organizing committee. If you've been involved with SeDNAs for a while, you'll know that we're going to have a really awesome conference coming up February 2027. So that means preparation for it now. So if you're interested in being part of that, please contact Aaron Hahn. And also, I'd like to highlight the CSIRO came out recently with the National Biodiversity DNA library. So the NBDL has launched. This has been a huge effort on a part of a lot of people in Australia, over 2,000 fish species. I'm hearing that people are reanalyzing their data and getting 20 to 30% more hits. Yeah, it should be a really, really great yeah, great resource for our eDNA community. And if you're able to see the chat, then you'll know that Ollie has posted in the NBDL link into the chat. But of course, you can also Google that. Now, we've also got a few webinar announcements. So webinar sponsorship is open. So if you're interested in putting your logo somewhere, this could be a cool place to reach a lot of people. Next webinar, tentatively on Azaiko and Martin, but I will be sending out a post-webinar survey to determine what month people would be available. As I know, January and February can potentially get full with things like holiday and vacation and seeing family and friends, which is also very important. Um, and then we will be searching for our 2026 webinar speakers. So if you got some really exciting results and can't wait until the next conference, then please let us know. We'd love to have you. And just letting people know that our past webinar recording is now on YouTube. So, without further ado, believe that Dr. Miwa Takahashi and almost graduated, almost newly minted Dr. Lara Denis-Roy will be talking about their research. I believe that Miwa will be talking and then we'll do some questions for her. So feel free to pop them into the chat or Q&A, and then we'll switch over to Lara. All right, ladies. Miwa, you be able to share? Yes, we can see CSIRO. Perfect.
Unmute. Can you hear me all right?
Yep, loud and clear.
Okay. Cool. All right. Thank you, Claire, for introduction, and then thank you for this opportunity to have a chance to introduce you about this newly launched IMOS eDNA sub-facility, which is really exciting. I'm very excited because this was recently launched mid this year, and then we're going to add a whole new layer for Australia's marine biodiversity monitoring. I would like to begin by acknowledging the traditional custodians and elders of land and sea on which we work and observe, and recognize them as Australia's first marine scientists and carers of sea country. So here on the right-hand side of the slide, there's a beautiful sea country artwork by Trunkin Singleton from Urunqangi, far North Queensland. And Trunking collaborated with IMOS to create this design, which reflects the way IMOS brings together Indigenous knowledge and modern science to deepen our understanding of Australia's marine environment. So with this beautiful artwork in our minds and hearts, I would like to pay my full respect to Aboriginal and Torres Strait Islander people, past and present. I'm going to tell you a bit about IMOS, which you might have not heard of, but it stands for Integrated Marine Observing System, and it is an Australian marine monitoring program. So IMOS is funded by Australian government and enabled by an NCRIS, and there are a consortium of institutions that you can see there listed. They have been operating IMOS with the lead agent of the University of Tasmania. So why was IMOS established? It's because oceans do matter to us as a blue economy in many different aspects. For example, national safety, sovereignty, security, energy and food security, climate change, and so on. And in order to sustain these valuable resources that ocean provides to us, we need high quality and sustained marine and climate data. So in order to provide this data and information, IMOS was established in 2006. So IMOS collects a huge range of physical and biological data from the coastal and open oceans in Australia. And all these data are open, usable, and reusable. All these long-term data sets are freely available for anyone, like scientists, students, industries, government, and any other stakeholders for their use in research and operational activities. And it will support assessments of oceans at both local, national, and global scale. So what kind of samples and data are collected and where are they collected? So IMOS has these 8 national reference stations that you can see in the map that are currently in operation. And these are not only the places where IMOS operates, but I wanted to especially highlight these sites because of their long-term history and also because these are where eDNA samples are collected from. So when I say long-term history, it goes more than 20 years of IMOS operation period. So three of these sites, Rottnest and Maria Island and Pohaki, these three sites have been operating since 1940s, so about 80 years. And at the same time, when these NRS were established, they started all sort of collections, data collections that you can see on the right-hand side. So these are the key data streams at NRS, not only, but some of the key ones. There you can see a range of physical and biochemical data streams such as temperature, salinity, turbidity, current, you name it, and biological and ecological data such as phytoplankton, zooplanktons, and the microbiome community data. So going back to the map, these NRS are located purposefully in important bioregions around Australia, and those bioregions are recognized to have different oceanic features, nutrient properties, and biological communities. So you can see this whole colorful, whole comprehensive full color pictures of ocean patterns here, which is really amazing. But there is something missing here, which is that it doesn't measure larger organisms that most people think about when they think about marine biodiversity. And this is where we are going to change with this eDNA sub-facility. So, Biomolecular Observing Facility is one of the 11 facilities that IMOS operates. And within this facility, there are three sub-facilities: Microbiome Coastal Sampling, Marine Microbiome Initiative, and this new environmental DNA sub-facilities. And I'm going to talk a bit about Marine Microbiome Initiative because they've been collecting the samples that we are going to use in our sub-facility. Marine Microbiome Initiative, it's led by Australian Microbiome Initiative and there's AM, Australian Microbiome Initiative. They collect eDNA samples not only from these NRS sites, but also from open ocean, fresh water. They collect soil, sediment, water. But specifically for this IMOS Marine Microbiome Initiative, they started their collection at the NRS on the East Coast in 2012, and then they expanded to the rest of the coast in 2015. They collect 2-liter water samples at the multiple depths from surface to the bottom. And the bottom can be 50 meters at some sites, or it goes down to 100 meters in some sites. They use Stervex filters and then extract the DNA using mobile kit with some modification. They apply 3 assays, 16S bacteria, 16S archaea, and 18S eukaryote. And what's amazing about this initiative is that it's not only about the longevity and the consistency of its operation, but also it's their commitment to make the protocols and data transparent and open. And as some of you may know, I'm very, very passionate about making eDNA data fair, and this initiative is really a role model in that space. So all their protocols from sampling to bioinformatics are very well documented and publicly available. All sort of data like raw data, processed data, very rich metadata, they're all also accessible through this bioplatform data portal. And they follow the international data standards such as MIXES. And so you can download their data by species or location or target regions. So I highly recommend to go to their resources and have a look at their documentations and then start looking at their data. And we are very fortunate to have their work as a foundation to build upon on our sub-facility. So here's another type of samples collected at NRS that is a plankton, phytoplankton and zooplankton samples. And I want to highlight this because we are also going to use this in our eDNA sub-facility. So at each site, at each time when they go out, so that's monthly at most sites, they deploy 3 drop nets, and 2 of them are preserved for in formalin for morphological identifications, abundance, and biomass records, and one of them is for DNA bulk samples. So DNA from all these existing samples have been already extracted, Some of them have been sequenced and analyzed in these two papers, for example. So these papers are from Tina Berry, who did a PhD at Curtin University. And in these papers, they revealed from the metabarcoding data, they revealed seasonal patterns, irregular patterns associated with extreme events like marine heat waves, and also detected endangered and invasive species. So these studies really showcase the power of profounding the real ecological patterns in IMOS data. And these are the types of data that we're aiming to generate at the larger time series. So eDNA sub-facility was launched in mid this year, and I joined the team very recently, but there have been years and years of discussions among Jody, Ollie, Bruce, and many other collaborators to build the ideas and then bring this project to this point. And the aim is to build upon existing and future IMOS eDNA samples and expand this long-term monitoring to macro, the larger organisms. And we are going to reveal a whole new extra layer of understanding about our oceans. Opportunities, there are a lot, and I'm really excited about these ideas and opportunities. So this data is going to have a huge extended space and time series. And also, they will come with a very rich metadata, including a physical and a chemical properties and a biological data as well, like a plankton counts and a microbiome community composition. So, with all this information combined together, there is a huge opportunity to run some modelings and then understand distribution patterns, understand how these macroorganism community compositions are impacted by multiple aspects, and also it can be used in biosecurity. I mentioned about this plankton biomass data that are already available, and I think it has a huge opportunity to address some of this eDNA quantification analysis. And then also we have we can improve, we can have a very good high accuracy of species identification thanks to MBDL. And of course, we would make the data fair at the not only within the IMOS database, but also across multiple study and multiple projects in this region, as well as at the global scale. So these opportunities that I listed here, I would like to highlight that it's important to understand that IMOS is a data initiative, and unfortunately, we're not going to analyze these data to do science. So we are funded to produce the data for other people to use and turn them into the real cool, fun science stuff. So this kind of data use cases are really important to demonstrate the impacts and the value of this data set, and so that we can continue to secure funding and keep on producing this valuable data set for other people to use. So please use eDNA data when it goes to live. So when is the data going to go live? You might be questioning. So this is our milestones that we've been working towards. Up until now, we've been engaging with eDNA practitioners and potential data end users to understand a few multiple things. For example, what are the priority taxa and the research interests are, what are the commonly used PCR assays in other projects, so we can make sure that we can try our best to ensure the interoperability of data across projects at the assay or gene level. And then also we want to understand opportunities and barriers that people might have in using IMOS eDNA data. So those informations were really useful for us towards the establishment of protocols, which is what we're working now. So we are running a few pilot studies to decide on assays, what assays we're going to apply, which is going to be a really important decisions to have the good representative coverage of species taxa that people are interested in. and also looking at the bioinformatic pipelines and that kind of protocols as well. So once we establish this, then we're going to process the samples, the old existing samples that are in the archive now, and make the data available hopefully around the mid next year. And after that, we have more activities that we want to do. So we're gonna, for example, continue engaging with the end users. And we want to review the protocols. Is the data accessible enough for them, even for those people without eDNA experience, for example? So this is a 2-year project to see what we can achieve. So we want to set it up at our best for other people to use the data. So we're really here to open the door to the community, to you guys, and or to anyone, and because we want to hear from you and how we can improve things on our end, then and then so we want to be involved and want to hear from you. So please reach out if interested. Okay, so this is the last slide. It's a take-home messages. We are leveraging existing IMOS frameworks and samples. We are currently working to establish the PCR onwards protocols. There are lots and lots of exciting opportunities in the data use, and we want the data to be used. So please reach out. With that, I would like to thank you all for listening to my talk and then receive any questions. Yeah, we are really here to hear anything from you, any questions, suggestions, or some ideas about data use or yeah, anything basically. So please reach out. Thank you very much.
Great. Do we have any questions? Yes, Ollie, I can see your hand. I've attempted to let you talk, Ollie. I don't know if that is working. Otherwise, feel free to type out your question and I guess drop it into the chat.
Sorry,
So,
I think that was an accident. Apologies.
We can hear you now.
Great talk, Miwa.
Thank you.
Just got a question for you, Miwa. Like, who have you been engaging with and what have they been saying? Like, what is it that people really want? What have you been seeing?
Yeah, thank you. So we started well, when I joined the team in mid this year, I started engaging with people that you would know, like people working in Australia mostly, because this is an Australian project. So I've been reaching out to the people who might be doing a more long-term monitoring using eDNA and understanding what kind of taxa they're looking at, what kind of assays they use. So I'm asking that kind of questions. And also we run end user survey. So we actually sent out the link, the SurveyMonkey link through the SeDNAs newsletter as well as the IMOS newsletter and other places. And we received really interesting inputs from there. So we ask questions like, what taxa are they interested in, what assays they've been using, and so that kind of technical questions, but also what type of data would you be interested in? And then all of them said they are really, they can use from raw data all the way to taxa, specific taxa distribution patterns. And then they're also interested in the plankton data, microbiome community. So I think there's a great interest in that. And what else? They also specify some specific interests that they might be able to address, and some like community, like about diversity assessment. Of course, that was not surprising, but some of them are interested in using to assess the ocean health indices and quantification analysis, use that into the government report, like a state of environment report. They've been already using IMOS data, but they are interested in implementing this new whole set of data from macro-organism perspective. Yeah, so those are the inputs that we receive and then really useful information for
Yeah,
us.
that sounds like it'll be huge. Any other questions from the audience? Give it a few minutes. If not, then Lara, would you like to start your talk?
Hi, everyone. Sure. I'll start. Okay, looks like it's working. Can everyone see that?
Yeah.
Awesome. Well, good afternoon. Thanks for having me as part of the webinar. I'm here to present some of the work I did during my PhD, which is essentially using eDNA for reef monitoring, comparing eDNA with visual surveys following the Reef Life Survey protocol. I did that work at the University of Tasmania. Some of you have probably already seen this presentation, so sorry for the repeat, but it's good practice for me, and hopefully you get some new information from it this time. I want to first start by acknowledging the traditional custodians on all the locations at which I was able to pursue my PhD, including where I am today, which is in Lutruwita, Tasmania, and so acknowledge the Muinina people and pay my respect to their elders, past, present, and emerging. Australian coastlines host some extremely biodiverse systems, such as the Great Southern Reef, who, with other ecosystems, are experiencing a decline in biodiversity. We can attempt to mitigate that with through monitoring efforts that we can better inform through the use of eDNA, which is a method we're all very aware of. It detects species present through what's left in the environment, and it's a method that can also benefit from a bit more standardization and validation at a continental scale for macroorganisms monitoring. This is what my PhD focused on. And so to further validate eDNA, I looked at detectability through this method, but also visual surveys following the Reef Life Survey protocol, compared that for day and night sampling and across very different bioregions, so tropical and temperate. To be able to do so, data was collected across 6 Australian states for a total of 54 sites, out of which 34 also had night sampling events. This is mainly due to whether or not we could go out at night to the sites and sample. A lot of those sites was just not necessarily the safest, or just not possible if conditions deteriorated over the next few days. So that's why there's quite a few sites where we were able to get night data as well. For the visual surveys, as I've mentioned before, we followed the Reef Life Survey protocol. Some of you might already be quite aware of that method, but essentially we deployed 2 divers along a 50-meter transect, and for a first swipe across a 5-meter belt on either side, all the kind of pelagic species, like your fish, etc., will be recorded, and then they come back and on a 1-meter belt from the transect record all the what we call more cryptic or invertebrate things that will be on the seafloor, like hiding a bit in your corals or in your kelp. And through that method, I was luckily able to witness a lot of the great biodiversity we have here in Australia, to try and tie back into my introduction, including at night, quite a lot of species kind of came out, which was really cool. At the same time as visual surveys were carried out, some eDNA was collected. So essentially, as you can see here in the catch bag, bottles that were previously cleaned out, decontaminated with bleach, were filled with water. But yeah, the bleach wouldn't be left in the bottle. We rinse it out beforehand. And to kind of align as best as I could with the RLS method, I used 2 assays, 16S fish and CO1 Lorraine. CO1 Lorraine was more to kind of capture that invertebrate biodiversity. here are some basic results to kind of give an idea. In green, we've got the eDNA, and in blue, the visual surveys. That's going to be the general color scheme. In total, eDNA detected just under 1,200 taxa across all sites, which is pretty impressive. Visual surveys were just underneath 800, so quite a bit less. And then if we look specifically at tropical and temperate, there is the pattern you'd kind of expect across both methods, which is slightly less taxa richness in temperate locations. The gap is significantly a bit more higher between the two methods in tropical locations. And for day-night, it's still the same trend. eDNA overall detects more taxa. Here, the lesser number of taxa detected by both methods at night don't have to do necessarily with the fact that it's at night. It's just that the number of sites sampled also at night is less than the number of sites sampled during the day. So overall, both methods had the same number of nocturnal species at around 130. However, for the overlap between tropical and temperate location, eDNA had higher overlap. After looking at overall taxa richness detection, I wanted to look at sorry, one sec, I've got things popping up so I can't see my own screen. At the level of resolution across both methods. And so overall, we can see that the visual surveys, thanks to the expertise of the divers, is able to detect taxa at a quite high level of resolution, usually species, at 96%. However, for eDNA, that percentage is quite low, or it's sitting at slight 72.5%, and quite a lot of taxa is only identified at the genus level at 22.6%. Also, it's only for eDNA that some of the taxa was identified at the infra-order level. For visual survey, the highest was usually family level. Infra-order for eDNA is mainly kind of your hermit crabs and your crabs. When looking at differences in resolution across tropical and temperate locations, there's a slight variation. UVC slightly loses resolution in tropical location, while it's the opposite for eDNA. eDNA gains a bit of resolution in tropical locations compared to temperate. And across day-night, we have kind of a similar pattern where somehow eDNA detects slightly better resolution at night, but not in a way that's really that significant. It's still around the 75% mark. For visual survey, there's like quite a bit of a drop down to 95% when it comes to night surveys with resolution at the species level. I also wanted to see the overlap between the two methods at different taxonomic ranks. So at the highest rank, which is class, sorry, the organisms that were identified by visual surveys by not the eDNA were a green turtle and a pycnogonid that were seen by divers but not recorded by eDNA. And as we kind of go down in resolution, we can see that that overlap shortens, like it's less and less. And so for the visual survey, that means about 388 species were seen by divers, but not recorded by eDNA. And I kind of wanted to look into why that might be. The first thing I did was check for reference sequences, and essentially about half of those, a bit underneath 40-ish%, were just, there was no sequences available. So eDNA was likely to have picked up on them, but just not get the actual species back. But another half did have sequences available. So to look a little bit deeper, I wanted to see if there was a chance that the taxa was detected, but not at the species level. So this is the overlay that you can see here. The different colors are the different, if a taxa has been detected at another resolution. So for example, we'll have like Oh, my brain's going. We'll have a hermit crab, and we'll have the taxa, so Strigiomanus. Yes. And the divers will identify it, but then eDNA, in the eDNA data, we'll only actually get back a Pagaridae or like a general hermit. And so for quite a lot of those taxa, another half of those, they're likely to have shown up in the eDNA data, but just not at the species level. All right. I next wanted to see if, in terms of assemblage, the methods kind of matched across location, or if they did. Generally, they did. As you can see here, in blue is the visual surveys, and in green is eDNA. There is some overlap in some of them that you can see, but it's mostly spread out. You have here the day and night, so it's exactly the same points, just overlaid depending on whether or not each sampling event or site was done during the day or the night. There's not much of a pattern. You can kind of see that it'll be more about location rather than methods being really different over day and night, but there's a bit of separation. Here are two great papers that have come out that look more into the day-night changes that can happen over reefs in Australia. Another thing that was interesting was the mean species richness was quite consistent across day and night in both temperate and tropical locations for eDNA. That's the purple kind of line that doesn't fluctuate too much between day and night. But when it comes to eDNA, when it comes to visual surveys, it's quite stable in the temperate locations. There's not a huge drop at night for the mean richness, so the taxa kind of number of different taxa identified on the survey. But when we look at tropical, there's quite a bit of a drop where the divers are really not picking up on the same amount of species at night or taxa. Now the part where it gets interesting is when I separate the temperate and the tropical locations. So here we can see that some of the tropical sites are in that case where the methods don't seem to matter too much and like drive and mean that there's like very different assemblages. Across temperate though, we can see like quite a bit of a differentiation between having sampled the same site with eDNA or visual survey. If we zoom in on the tropical sites, we can kind of see that the method was as important as the location, whether it was the GBR or North WA. While if we look at eDNA versus visual survey in the temperate locations, it's really that the method that drives that big difference. One overlaying kind of the species that drive that difference, well, we can see that it's kind of the same for the tropical, but for the temperate sites where it's quite interesting, these eDNA is picking up a little bit more on some of the pelagic species like the Trachurus or the Sardinops sagax. And for visual surveys, it's more the inverts. So the like little turbos, or I think now they're called undulatus, or your abalone, Helvetius rubra, and urchins, Helucidaris erythrogramma. So overall, what I've kind of found out is a lot of the taxa missed by eDNA is really a matter of reference sequences, which is with the launch of the NBDL is a huge help in that area. Probably going to solve a lot of the issues of just having taxa that are seen by divers but not really picked up by eDNA. The also other interesting thing was that there was not really a strong nocturnal pattern across the eDNA samples, which is kind of good news. It probably means that if we do one sampling event, it kind of carries through that 24-hour signal, at least in like most of the sites that I've done. There is, however, in terms of how well the eDNA method performs, a big difference between if we're in a temperate or a tropical location. For specific examples, in Tassie, we do have like a lot of leather jackets and cowfish, and divers will be able to differentiate those quite easily, as you can see in the pictures. But for eDNA, it would usually just stop at the genus level and not be able to bring that resolution to the species level. So another limitation on the other side for visual surveys that we kind of saw with that drop in mean richness at night was probably more of a diver visibility. If we're out at night with lamp torches, we're kind of having like a very narrowed vision of what's on the reef compared to during the day where we can very much see what's around us. Yeah, as I've mentioned, the National Biodiversity DNA Library Guide is getting launched, and that's great news. It's going to really help bring eDNA further into its full potential. And then there's also the quantity side of eDNA. That's one thing I'm hoping, well, I have looked into and I'm hoping to refine by comparing biomass and abundance that we get from visual surveys to like the eDNA data. Here are the key takeaways I've kind of gone over during the discussion. And I also want to say a big thank you to all the people that have helped me pursue that PhD. And this project was massive and it wouldn't have been possible with all those wonderful people. So there you go. Thank you very much.
Great. That's been a really awesome talk. Looks like you've done quite a lot of work. See, we're getting some clapping and hearts and stuff in
Thank
as
you.
little reactions. I think Ollie's asked a question around sort of, I guess, repeatability. So he's saying that, a useful feature of eDNA is that we can sort of review identifications and improve on them, as you've mentioned, I guess, with the MBDL coming out. So unless observers took photos, maybe it's harder to reanalyze diver surveys. Can you just talk a little bit more about like how you guys did diver surveys and if there was like repeatability and like replicates and things? And
Sure.
like, was there like variability in that?
Well, there's a little bit of variability in the level of resolution. Trying to get to this slide.
Sorry to make you go all the way back.
No, all good. There's that little box kind of shows the method, but I might have not mentioned we actually record down. That is probably a very good point. We record down on a slate the size and the species and how many there are along the surveys. A lot of the time, if anything's not identified pretty much right away, we have like cameras and we take pictures. You can kind of see it on that picture of me. I've got the catchbacks for eDNA and I've got the transect line, but I've got also a camera. And that's
Cool.
for all the kind of, the more of your cryptic. If you get a bit of a weird looking weedfish, you can take a picture of it and then figure it out. Yeah, I think that was the question. Did I miss a part of it?
Yeah. And I think there's Martin's asking sort of a follow-up to this question. So I feel like I'll just tack it on to this one. Is there any way to look at like potential diver observer bias? Because it seems like all of your divers obviously knew a lot about the taxa that they were seeing, but potentially if like a novice diver went in, would there be maybe more variability in the data?
Yeah, that's a good point. RLS does quite a bit of training, so there'll be like training weekends and different moments where people upskill. For example, when I first started, I was doing just what we call method 2. It's kind of your cryptic species, like just a 1 meter belt and then looking through the kelp or around in the corals. So we kind of build that skill up to get better until we have a level of confidence where we can, even if we can't get necessarily a good picture, we can have a good sense of what family or what genus taxa we've seen. But in terms of, definitely in terms of bias at night for night surveys, there was, that was definitely something I didn't look a lot into, but that showed up kind of in the graphs I showed where if we compare, it's just, I think it's with the torch, the fact that also will be on the reef and there's this light moving through the reef, there's probably a lot of species that will be shy and won't necessarily want to come anywhere near us. So we lose quite a bit of information there. And it's also something that we can that can happen as well with surveys that are on transect lines is because we reel out the transect and then we record fish and invertebrates, that first reel out often will spook a little bit some of the fish away. and they might not come back right away. And so sometimes they don't get recorded on that method one, but we try to record them as like a method 0, especially if they're quite unusual, or if it's a little wrasse and we've recorded lots of wrasses, it doesn't matter so much. But if it's a whole other species, sometimes it's worth noting down. Yeah, at least that's my insight into some of the bias associated with diving.
Yeah, totally. Andrew's got a question. He's saying, what are the natural processes such as degradation, transport, behavior, etc., that could explain some of the patterns that you saw?
For eDNA, I imagine.
Yeah.
Yep. It was tricky. There is definitely, I thought, I didn't do it, but I did have a think about potentially looking into how long it took me to filter the samples. It could happen pretty quickly. Some of the sites, especially tropical, if I didn't have access to ice and I couldn't filter right away because we were on a boat, sometimes I lost quite a few samples, actually. I did a lot more than 54 sites. But a lot of the sites, just sometimes it doesn't work out. I couldn't filter in time, or the water would just be exposed to the sun, even though I tried to cover it up. So degradation definitely had a big part, probably in some of the quality. I can't exactly measure it in the case of my study, but I did. It is something though that I write in the methods to kind of explain that, like, yeah, we filtered it sometimes, yeah, within 24 hours. But sometimes it would be a few hours because of just logistics. Ideally, though, if you could filter right away on the boat or like at the dive site, I'm sure the quality of the data would be increased. It would be really interesting.
So did you use any preservative in your
No,
sample?
just freeze them.
Wow.
Yeah,
Cool.
I would not do that again. I would probably get preservatives.
Yeah. Ollie asks, hi, Lara. Given that in some cases eDNA and observations are picking up different components of the biota, how can we carry on eDNA surveys from long-term visual surveys? Seems tricky to maintain continuity, so any ideas how we could tackle this?
Yes, I don't see it necessarily as a problem. I kind of think like visual surveys are great, and they have, like in the case of RLS, they've been around for a long time. So it's this really interesting data set with over 20 years and sites all over Australia and all over the world. So kind of it's actually part of potentially my future postdoc, is further aligning eDNA with RLS. So whenever people go out and do some RLS surveys, they'll start incorporating eDNA sampling. I think I might not fully understand the question. I do know that eDNA is going to pick up on different things, and sometimes it'll be a lot smaller just because, for example, for RLS, we don't go under 2.5 centimeters, but there's quite a lot of taxa, especially like on your corals or like kind of your tiny invertebrates that will be smaller than that at certain stages of their life, or like fish as well. So they complement each other. I wouldn't really choose one over the other. I kind of think they highlight different parts, but they can inform quite well on the reef separately, but also joined. If that makes sense.
Yeah, I think there's a lot of discussion in the literature as well about how eDNA isn't necessarily a replacement for, I think, other methods.
Yeah, exactly.
Yeah. And then Andrew asks a question, how confident are you that the difference is biological, I guess, between the methods and not an artifact of how the data was collected or the samples were processed?
Pretty confident. It still aligns quite well. We still have a lot of species that come up in both methods. And then looking into, I mean, I'm pretty confident. I try to check it best I can, but I'm also very open to suggestions. But yeah, there is quite a bit of overlap still between the two methods. And then when I was kind of trying to check which taxa were not detected by eDNA, it was mostly a matter of no reference sequences. We tried to sample, I mean, there is little ways we can definitely optimize. The eDNA samples were getting taken at each end of the transects, just in terms of logistics with divers, making sure everything could kind of smoothly run. But I do imagine though that like if 1 liter samples were taken at like every 5 meters or such, there might be slightly more species that would get picked up, but I don't think it would be that much of a difference because there was also that consistency between day and night sampling events where the eDNA didn't have that much of a variation at the same site. Well, for visual surveys, there was quite a bit of change on what was detected at night. So I think they were as closely as they can be with, being realistic in terms of organization and what's doable. But there's probably, I think maybe what would matter more is which assay was used, like especially in terms of invertebrates. If instead of CO1 Lara, I chose other assays that might have had a better resolution, then the data would have looked quite different in that sense for eDNA.
Yeah, that sounds pretty reasonable to me. Do
Yeah,
you think
not
that you
all.
would
Mhm.
do you think that you would get more of more hits, I guess, with your eDNA with the new national database? Do you think that would actually make a difference?
Yeah, I think probably there'd be quite a lot of species that would show up that didn't show up before, and not just the species that are necessarily detected by UVC, but a lot of your more pelagic species that go over reef. I think
Oh,
quite a bit will get picked up because eDNA was a great data set full of surprise. I still remember the day I found out that there was a mola that had been picked up by at one of the sites, and I got so excited. So, but yeah, I think a lot more would get picked up.
yeah, well, that's really awesome. Well, hopefully one day you can go back and reanalyze with a more full database.
Definitely would love to.
All right, so those are our 2 speakers for the day. And if we could just give our speakers just another big round of Zoom reactions to thank them for sharing their knowledge with us. It's been really wonderful, guys. I'll hang around if there's any questions or anything else. Otherwise, have a great day.